It's also my pleasure to introduce the next speaker, Jennifer Pett-Ridge. And I take a special pleasure in this because, for me, she's emblematic of the type of person we're trying to support with a building like this. She is a senior staff scientist at Lawrence Livermore National Laboratory. She got her PhD here at Berkeley with one of my scientific heroes, Mary Firestone, who is responsible, I think, in many ways for us appreciating the depth and beauty in the microbial ecology that surrounds how soil operates and plants operate and interoperate. But very importantly, what Jen brings is a set of different types of data that track dynamically how materials flow through these systems and are dynamically reallocated and reused to create the mineral cycles of the earth, which transform our soils, that make our plants grow. It's the technologies that she brings that make this at all possible. What KBase is supposed to do is enable her to turn her data and that sequence into the most sophisticated models of biological function we've ever had. So Jen, I look forward to what you have to say. Thank you.
Thank you, Adam, for that introduction. Thanks. I'm really honored to have been invited to speak today, and I'm speaking both as the lead of a team of folks at Lawrence Livermore Lab who utilize the resources that are generously provided by the JGI and by KBase and the Genomic Sciences program in Washington. But also, I'm here to just reflect on a little bit of what is the user experience, hopefully not the over-user experience, but we take advantage of the data, the resources that we get from KBase and the JGI, but also the partnership, because it's not just a one-way conversation. We really do partner with folks in Walnut Creek and here in Berkeley. So in a nutshell, what our group does is what I call community systems biology. That's perverting maybe the original way we use the term systems biology, but we're trying to understand ecological patterns of complex microbial communities, specifically ones that are involved in fostering the productivity of organisms that might be in an algal biofuel pond or in a field of switchgrass, so they're relevant to bioenergy in some way. And we're interested in these interactions because they have an ecological foundation. It's predator-prey interactions. It's regulation of nutrient and energy cycles. It's shaping the environment that the bioenergy crop has to grow in. And while that's important for growing bioenergy crops, it's also just really fundamental microbial ecology. And I'll have to say that that's kind of where my motivation comes for. So to do this work, we need lots of different kinds of data. And we need sequence data. We need information on transcripts and signaling. And we need to be able to put that into metabolic models to be predictive, as Adam just suggested. We, in my group, happen to use stable isotopes because those allow us to track specific molecules as they move through plants and into the fungi and then the bacteria and then maybe into phage. So they allow us to track those energy and nutrient exchanges in these systems. But if we were to have to generate all of that data ourselves, and probably more importantly, if we had to come up with the methods to achieve that data, if we had to learn how to do enrichment of messenger RNA, if we had to learn how to close and annotate genomes, if we had to make all of our own flux balance analysis models, we wouldn't get to do science, what I call science. We wouldn't get to answer the ecological questions, which I think of as being our core mission. So that's where our partnership with the JGI and KBase comes in. And it's not just to develop methods, it's to do informatics and understand the data that we're being provided as well. So I just want to tell you a short scientific story about how we've used some of the data that we've gotten from these resources in an actual scientific context. Okay, so here we are in the root zone of an annual grass, Avena barbata. This is surrounding, if you go out into the grasslands here at LBL, you'll see it all over the place. We are interested in understanding how roots like these stimulate the genes that are involved in decomposition in soil. The area that we call the rhizosphere, that's the area that you can see it right there. It's those little root hairs, it's within a couple millimeters of the root. And if you're living as a microorganism in soil, bacteria, archaea, fungi, doesn't matter, you're living in a little bit of a nutrient-limited wasteland of an environment. You may have the ability to go after some old carbon organic matter that's there, but it is not an energy-rich source for you until this root comes cruising by. And then all of a sudden, there are exudates, there are low molecular weight sugars and amino acids and organic acids, and that is just candy, metabolically speaking, for that microorganism. Okay? So we get this great growth of microorganisms and activity. But what we know at the ecosystem scale is that root coming through actually causes loss, respiration of the pre-existing old nasty, carbon organic matter that was in the soil, that was not yummy enough to eat yesterday, but for some reason it is today. And we call that the priming effect. And it has global implications, right? Because we are doing an experiment on this Earth right now where we are warming and we are increasing the CO2 level, and that causes that priming effect to increase, at least when we do the experiments at the ecosystem scale. So what are the microbial and genomic underpinnings of that phenomenon? So we did an experiment where we grew this Arena barbata grass in soil. In some of the treatments, we had old, dead organic matter, some we didn't. And we looked at the bacteria and specifically at their gene transcripts, which are an implication of what kind of activities they're up to in that rhizosphere zone. And my postdoc, Yara Nuccio, took the transcript data that had been sequenced at the JGI, and she mapped it to a massive metagenome that also came from some JGI data that was produced by Owen Brody's group. Owen's here at LBL. And she found she was able to map a number of genes from specific organisms. So this is a Jathinobacterium. It's a bacterium that lives in the rhizosphere. And she saw upregulation of genes that we call CASI, or carbohydrate active, genes that we know to be involved in degradation and decomposition. Upregulation, there's a pattern with time, high at the beginning when the root first grows and then declining with time. But upregulation of these genes in the rhizosphere. So this is an organism who lives near a root, but chooses to eat spinach when there's candy everywhere, right? It's choosing to eat complex macromolecular organic matter and break it down when it could be eating sugars. So why is that? We think this is the kind of organism that underlies the priming effect. So there are very, very few. While there are we found 8 genomes out of everything that we sequenced, there are relatively few organisms who are really doing this activity, though a lot of organisms have the capacity. So to really get at why it is that this organism, or what it is that this organism is doing that all of the others who are very much eating those sugars produced by the root, we are using something called stable isotope probing. And I don't want to go into too much detail, but essentially we grew those plants initially in an environment that was in a box that was filled with 13 CO2. So those exudates that the plant produces in the soil are labeled with the heavy 13 CO2 signal. So if an organism is taking up those exudates, that will be incorporated into their DNA. If they, on the other hand, are in another treatment where they're taking up 13C labeled decaying litter, that will be incorporated into their DNA. And if we both separate out that heavy DNA, you can see a centrifuge tube here where we've actually physically separated the heavy DNA and the light. We can sequence that, we can do metagenome sequencing of that, and really hone in on a particular taxa who prefer to eat this versus that substrate. Now, this is a method that's been around for 10 years. Folks use it all over the world. It is labor intensive. I could use another word. It takes a long time. You literally are dripping, dripping, dripping slowly individual fractions of DNA. We've partnered with the JGI, and we're very thankful for the Emerging Technologies Opportunity Program to automate this, to make it faster, 3 or 4 times faster, but more importantly, done by a robot, so that there does not need to be a technician in the lab doing this. We're then going to send the separated DNA off to the JGI for them to do metagenomic sequencing, and that's going to tell us the whole community of organisms that are involved in eating this, what I call candy, involved in eating these more complex substrates, which really gets at that mechanism of priming. So we're very thankful for the opportunity to work with the JGI and develop this method. This is certainly not something we could have just sort of financially come up with the wherewithal to do without the support that they're generating for us. And I want to just leave you with one other very sort of personal note of a new project. There's no data for yet. We've just done the experiments. But I was blessed to receive an early career award from the Department of Energy a year and a half ago. And the JGI was willing to go out on a limb and fund a community sequencing program grant to support the sequencing of transcripts from mucky, clayey, tropical soils, which are very difficult to get RNA and DNA out of. But these are the soils that could be significantly impacted as our climate changes. And we look at carbon, that huge amount of carbon that's stored in these tropical soils, and we know almost nothing about those microbial inhabitants. So I just want to say I feel very blessed, as it were, to have been the recipient of these partnerships and this data and this opportunities. And really the program that we've developed supporting DOE missions really could not have happened without these generous support. So thanks.